So this is a typical prokaryotic gene. It's called an operon. Okay, you've got the promoter, you've got the operator, and then you've got five, in this case, 4 coding regions. Now each of these coding regions literally codes for a completely separate protein. One of the ways eukaryotes are different is each promoter is associated with just one coding region. These aren't exons, these are actually completely separate coding regions. It's done this way in prokaryotes because these different genes, these different proteins that are produced are all involved in the same process. So it's very efficient if you can turn them all on and all off at the same time. Okay, so that's the structure of a prokaryotic operon. Okay? So we have the promoter for RNA polymerase binding, the operator for the regulatory protein. For worksheet number 2, we're working on an example where we're assuming that this operon is regulated by a repressor. Okay, so we have a repressor protein that can regulate expression of this gene. Okay, for the first part, I want you to describe, okay, well, how would a repressor actually function? How would it act to turn off expression? How does it actually work? And the second part is a little bit harder. How can you actually use just the repressor protein, no any other additional proteins, just the repressor protein to actually turn on expression? How can you use a repressor to turn on expression of this gene? Okay? So work on that for a couple minutes. And think about this. So how do you use a repressor? How does it work to turn off the gene? And how can you actually turn a gene off? It attaches the operator. How does that block gene expression? How does binding the repressor block expression? What is involved in the expression of the gene? What's the first thing that has to happen? What's the first thing that has to happen in expression of the gene? You have to transcribe the RNA. That involves what? What does that? RNA polymerase. RNA polymerase has to bind to the gene and then read through and transcribe it, right? So how does a repressor block that?
It blocks the RNA polymerase. Or it traces.
It attaches the operator and it blocks the polymerase, right? It can either prevent it from binding the promoter in the first place, or even if it does bind, you've got the repressor sitting right in front of it and it can't get past it, so it gets in the way. That's one of the ways repressors can often block expression.